hydrophobic signal sequence is recognized by a signal recognition particle, SRP. SRP binds to both the signal sequence and the ribosome. Resting further translation. SRP ribosome nascent polypeptide binds to cytosolic surface of ER membrane. SRP is released from its ER receptor and the ribosome becomes attached to translocation channel. Signal sequence on nascent polypeptide is inserted into narrow aqueous translocation channel, displacing plug within. Growing polypeptide is translocated through the hydrophobic pore ring into ER lumen. Translation is terminated and completed. Polypeptide passes through translocation channel. Membrane-bound ribosome is released from ER membrane and helical plug is reinserted.
Glycosylation and protein folding happen next within the ER lumen. Oligosacchαρyltransferase adds a carbohydrate chain from the dolichal phosphate to the polypeptide.
Molecular chaperone bind terminal glucose residue to the glycosylated polypeptide. The chaperone ensures proper folding. Disulfide bonds are created by the protein disulfide isomerase within the protein. By removing one glucose molecule, the chaperone connects and checks the protein. If misfolded, another glucose is added and then brought back to the molecular proteins to be folded correctly. If the protein is no longer needed or has to be removed, mannose is removed and is then brought to the proteasome. When folded correctly, it is moved to the cis-Golgi via transport vesicle. The cis-Golgi either moves the protein to the cisternae or back to the ER lumen. From the cis-cisternae, it is then moves to the medial cisterna, then to the trans-cisterna. From there, they are put into vesicles and sent to their destinations. Glycosylation occurs at the end of the trans-Golgi.
The polypeptide is moved to the lysosome by the transport vesicle. Tethering occurs between the two RABs. Docking is facilitated between the V snare and the T snare by the tethering protein. Finally, fusion occurs when the V snare and the T snare generate enough force. The protein is then delivered.